All right, welcome to Life and Biology. I'm Dr. Joel Graff, and this is a condensed lecture for a presentation I made on January 21st, 2021. I'm just going to review the slides that we went over in my Principles of Living Systems class, and the first thing I noticed is that I spelled swimming wrong, so let me change that. And, okay. We're just finishing up water, and we're going to move on from water to functional groups. So a couple of things to know about water. It can be a gas, a liquid, or a solid. In the gas form, it's water vapor, or you could say humidity. As a liquid, we just call it straight up water. And when it's a solid, it's ice. Now the liquid form is important because that's typically what you'd find in cells, and all molecules that we've been discussing and will discuss are basically swimming in water. So you got to think about the properties of water and how your molecules will interact with the water. And a really important property of water is that it has a high specific heat, and it also takes a lot of energy, which means it takes a lot of energy to change the temperature. This is important because it provides stability in environments. Because it takes a lot of heat or a lot of energy to either be released to lower the temperature or added in to raise the temperature, it keeps the living things' bodies at a constant temperature, and in the form of humidity, it keeps the temperature from doing large fluctuations. And finally, living systems don't tolerate environmental changes very well, and so between being able to maintain an organism's body heat and the ability ability for humidity to keep temperatures from changing rapidly, all those work together in the favor of life. So that was it for water. The main topic today is functional groups. We've been discussing hydrocarbons, and those were long chains of carbon and some hydrogens, as the name might imply. However, we can I'll find all the spelling typos I made in class. We can make more complex carbon-based molecules, and basically you could think of it as decorating the hydrocarbons that we've been learning about with functional groups. Now, even before we get to hydrocarbons, if we just draw out a water molecule, we could see that you could break one of these bonds, and if that were to happen, you would end up with a hydrogen ion, also known as a proton, and then an OH group with a negative charge. And so the OH group is going to be similar to a hydroxyl, so you can kind of think of water as a hydroxyl with a proton added on. So in cells, it's common for the pH to be 7.4, and when molecules are broken down or release protons, that can cause the cells to become more acidic, and so we have buffers within our cells to prevent large swings in pH. And then just as a side note, if blood becomes acidic, we just breathe harder and use the carbon dioxide being carried by the blood as a buffer. And we'll talk more about that at a later time. Okay, so back to the hydrocarbons. We discussed how methane has 1 carbon, ethane has 2 carbons, butane has 4 carbons, and then we saw that if you were to draw out butane in shorthand, we would have a zigzag black line. Then I drew in red dots to indicate where the carbons are on the red lines, and then I was being lazy and I just drew blue dots showing where the hydrogens would be. But anyway, if we counted up all the blue dots along with those 4 carbons, the formula for butane is C4H10. However, I wanted to point out in class that there are things called isomers, and that means that we can have 2 different types of molecules that are butane, However, we need to be more specific when we discuss butane when it's rearranged. So isobutane has a formula of C4H10, but it is the carbons and hydrogens have been rearranged. So it still has the 4 red carbon dots on here, and it still has a total of 10 hydrogens floating around it. But you can see that the butane structure here and the isobutane structure here are different. And so when we learned about the honk rule, we learned that I had told you that I could give you the chemical formula of something, and you should be able to figure out whether that what the structure of the molecule was based on that chemical formula. But this is a little different once you get to larger and larger molecules. But the honk rule still comes in handy because we could go in and check out these molecules and make sure that each carbon is still associated with sharing 4 bonds, and each hydrogen has 1 bond. And that was true for both of these molecules that we drew out. So all this means is that we had to come up with some nomenclature to describe alternatives to just straight carbon chains that we had learned about. Okay, so let's give another example of when you might not have just a straight carbon chain, and that is hexane. Hex for 6 carbons, and then ane single bonds. So I just drew a zigzag line, and we could number it 1, 2, 3, 4, 5, 6, and those are the 6 carbons on hexane. And then we could also draw a circle version of hexane. So 1, 2, 3, 4, 5, 6. There's 6 carbons in this molecule as well. And then you can also count the number of hydrogens. There's going to be a different number of hydrogens in the circle versus the one that's closed up, I think. Actually, let me count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, so there's, this was C6H14. And this one is C6H12. And the reason is that some of the you have more carbons interacting. This carbon on the end here interacted with the carbon on the end here. Okay, I'm getting off topic. Circle. So circle hexane, we can call it cyclohexane. Now I wanted to talk about the fact that you could also have double bonds within a cyclical carbon molecule, and we're going to see more of these cyclical carbon molecules coming up. For example, when we talk about steroids, they're going to be made of a number of 6 and 5 carbon circles. Okay, so anyway, double bonds can occur in the circles, and there's a special type of 6 carbon circle molecule that I want to talk about, and it goes by the name of benzene. This is a hazardous material. It has the six carbons, just like the regular hexane had, but then it has double bonds alternating between each one. And then at this point, the chemical formula is C6H6 because each carbon only has a double bond to another carbon, a single bond to another carbon, and a single bond to a hydrogen. Okay, so one thing about all these electrons that are being shared is it's not always twice as many electrons here as it is here along the right side. So instead, those electrons are going to be essentially moving around that circle equally. And this little thing that looks like a nut from a bolt, it's a hexagon like we've shown before, and then you just draw a circle in the middle, and that is how you can do a shorthand version to explain what benzene looks like. Okay, so this page has a lot of writing in it, and so I'm just going to take it easy going from the left side to the right side. So at first, I just drew this structure here. It happens to be the amino acid cysteine. And I drew all the carbons and nitrogens and hydrogens. And then you can see later on, as I was talking through the slide, I kind of pointed out that there's a group of atoms here with a certain pattern, another group of atoms down here. And although this structure might be kind of overwhelming when we just started talking about chemistry 2 weeks ago, the main point is as molecules get more complex, if you think of them, if you start to understand some of the functional groups, then it's just a matter of noticing that, oh, we have a carbon in the center with a carboxyl group to the right, an amino group to the left. There's a single carbon here with a couple of hydrogens, and then this ends off with a thiol or a sulfhydryl, two different names for the same thing. Okay, so this particular molecule has three different functional groups, but over on the right side of the page, I'm going to draw out. I drew out 7 functional groups that we should know in class. So if I were to ask on a quiz question, you should be able to know that that how to draw each of these 7 types of functional groups. Ignore this blue part for now. I'll come back to that at the end. So let's start here with the methyl group. The methyl group I wrote, it looks a lot like a methane, except for in a methane, instead of this R, there was an H there. Now R can be shorthand for anything, is my not very technical way of describing it, but an R is, it can be whatever atoms else is in this molecule. So we're just kind of shortening it up so that we can focus on whatever is connected to the R. And in this case, we've got a carbon with 3 hydrogens, and it's called a methyl group. In this functional group here, called a hydroxyl, R is connected to an oxygen, which is connected to a hydrogen. And the oxygen wants to form 2 bonds, and so it forms one with hydrogen and one with the R group. And then each of those shared bonds represents 2 electrons that the oxygen feels like it has. And so I drew in a few more dots. I don't know why when I save my PowerPoints, all my drawings get kind of messed up. But those originally started out as individual dots. So all of these slides are kind of messy looking. And it's half of it is my handwriting, but half of it is the change that occurs as you go and save a file that you've been drawing on a smart podium. So excuse the occasional craziness. We talked about a sulfhydryl group or a thiol group over here on this amino acid, the cysteine that we drew earlier. And so if we're going to just give it an R group to stick onto, we'll have an R with a sulfur and a hydrogen. Again, this is just supposed to be 2 pairs of electrons sitting there on the sulfur. Sulfur sits right below oxygen on the periodic table, and so if oxygen likes to form a configuration like this, sulfur will also do the same thing because it's in the same group. So there's a lot of similarities between a sulfhydryl and a hydroxyl. Again, my electron dots got kind of all weird on me, but here is another functional group called an amino. And we have an R group, a nitrogen, and a couple hydrogens. And you can count that there's 3 bonds coming out from this nitrogen, and that works with our honk rule that we learned. And then a couple extra electrons that I sometimes don't draw. Moving on. Let's look at this one. It's a carboxyl, because we had a carboxyl over here that we talked about. I drew a little dotted line around this part of the carboxyl group and this part of the carboxyl group, but a carboxyl is attached to an R. You've got a carbon with a double bonded to an oxygen, and the carbon also has a single bond to an oxygen and a hydrogen. Okay, so there's a lot going on with the carboxyl, but I've got these blue arrows pointing because what this carboxyl is a combination of carbonyl, which we haven't talked about yet, and hydroxyl. And even if you take the portmanteau of carbonyl and hydroxyl, you end up with carboxyl. So hopefully that is kind of a way that you can keep straight which one is a carbonyl and which one is a carboxyl. Okay, so the carboxyl, C double bonded to O and an OH. Okay, so the carbonyl portion of the carboxyl can be described in our R method with an R1 here and an R2 over here, so that this carbon that's in the center is bonded on one side with an R group and on the other side with a second R group. Okay, and then there's double bonded to an oxygen. So here we can see a carbon double bonded to an oxygen. There's one of the R groups, and then this OH could be considered a second R group. And so that is how we've got a carbonyl here, and then the hydroxyl is pretty easy to see here. And so anyway, we only have 7 functional groups to remember in this class, and 2 of the functional groups can be merged together into a third. The sulfhydryl looks just like the hydroxyl. And then finally, we've got kind of an oddball one here, and it's called a phosphate. So we've got an R group bonded to an oxygen, bonded to a phosphorus, and then this phosphorus is doing something weird that we haven't seen before. It's got 5 bonds going on, and then it's got kind of a hydroxyl group up top and another hydroxyl group and a double bonded to an oxygen. And so this is just totally oxygen loaded. And if we remember that oxygen is electronegative, that this is could potentially have a negative charge. Okay, so there's walking through each of those. I would recommend just making some flashcards or something along those lines, or just drawing these out by hand quite a few times, because once you can spot functional groups, then the larger molecules we look at, such as this amino acid over here on the left side, is going to be less intimidating. Okay, now speaking of those oxygens being electronegative, what I had drawn here, and it's messed up by when I saved the file, is that the electrons, the 2 electrons that were forming a covalent bond between the oxygen and the hydrogen, the oxygen got a little too greedy and it pulled the electrons completely away from this hydrogen. And a hydrogen is a proton and an electron. So if you take a proton away from a hydrogen, you're left with just a proton floating off. So although I write H+, you can think of it as a hydrogen ion, or you could just call it a proton. So I'll be doing we'll have lots of H+ floating around in for future discussions. And so if I remember, I will probably say proton most often, but I might say hydrogen ion. So just be ready for any sorts of slips of the tongue and changing around of nomenclature as I'm trying to go through this chemistry with you guys. So I'm getting a few chats and I don't understand what they mean. I'll see if I can shut off the chats in a future episode. It looks kind of Russian to me. So just ignore the chat, I guess, is what I'm trying to say. Okay, so because we had just talked about how the hydrogen can lose the electron battle with the oxygen and a proton pops off, we end up with a charged molecule, because if the plus charge goes away, you're left with an extra electron here, and so it has a negative charge. So that's an example of a charged functional group. And so of the 7 functional groups we talked about, 3 of them will often have a charge under biological situations, and that's at that pH 7.4 that we talked about. So just a quick message, or here I drew the carboxyl again, and this started out as an uncharged carboxyl, but again we have this is an electron, this is an electron, that electron pair, the oxygen got too greedy, and then the hydrogen without an electron is just a proton, and it popped off. So then this is what it would look like afterwards. It would have a -1 charge, and we'd put a little minus sign next to that oxygen. And then this is something I went over in class. I kind of regret it now, but I was basically trying to point out that just like on the benzene, where the electrons are fluttering around here and here and here, at this point, this is fairly symmetrical, and the electrons will be more or less evenly distributed. So if you were really to be correct, you would have just kind of a 1 negative charge kind of just around this area. Okay, well, the amino group is the next one we're going to talk about. It has we have the R with a nitrogen and a couple of hydrogens. In class, I did a nice job of pointing out that it has a couple electrons there, but of course it got messed up. So then we've got a hydrogen here, and nitrogen's fairly electronegative, so it's going to have kind of an ability to pull the electrons in a little closer there. And then this hydrogen's also going to be losing the battle, and so we've got quite a few electrons floating around this nitrogen, which gives us a bit of a negative charge. It's only partial, but you got a bit of a negative charge, and so some of those electrons could then form a bond with that proton that was floating around, whether that proton came from this part carboxyl like I drew it, or just happened to be somewhere in the water. That happens. So when all the dust settles here, we've got an R group, a nitrogen, and then it has 3 hydrogens. And so then there's just in general a positive charge amongst this whole functional group. So there's a +1 charge. So we got a -1 charge and a +1 charge. The most extreme example we'll go over is the phosphate group. And again, this is the bigger one. There's more to draw, but you've got an R group connected to an oxygen. There's a phosphorus in the middle here, and we've got a double bonded oxygen, another single bond to oxygen, single bond to an oxygen, and each of those single bonds to an oxygen, those oxygens would like to form 2 bonds to be happiest, and so they form a bond with hydrogen. But just like in this case over here with the carboxyl, where the oxygen won out and pulled off the electron from the hydrogen, that would happen here in kind of this hydroxyl group looking thing, and also it would happen here in the other hydroxyl looking thing. So in this case, we have 2 protons pop off the molecule. So then without those hydrogens drawn on there, we've got an R group, there's an oxygen, a central phosphorus, double bond to oxygen, and then each of these oxygens here are going to be negative charged, but of course the electrons are going to be all flowing around, so you just kind of have a general overall -2 charge around all these oxygens floating around this phosphorus, so -2 charge overall. And let's see, amino acids is the next thing we talked about. We already covered one amino acid, but I wanted to draw an amino acid that could end up with a charge. So we start off with aspartate and aspartic acid. And so what happens here, I'm drawing the amino acid. Here's our central carbon, which is sometimes called the alpha carbon. and it has a carboxyl group to the right, an amino group to the left, a hydrogen up, and then down here is what's called the R group. And so aspartate's different than cysteine, and the difference happens to be in the R group. In fact, all 20 different amino acids that we will consider in this class, they all have a different sets of atoms down here on the R group. So in this case, it starts out looking a lot like that cysteine that we drew earlier, where the R group has a carbon with a couple of hydrogens. But then where there was a sulfur group on last time, we've got a carbon group, and it turns out, if we take a quick look, we could see that this whole thing here is one of those carboxyls that we've written out a few times so far. We know that this hydrogen here might lose the battle of the electron tug-of-war with the oxygen, and so a hydrogen without its electron is a proton. So as we move to the right in this chemical reaction, the proton would pop off, and you would have and if we go from the left, we would have a proton going on. Now, I drew this version of aspartate. I think this is aspartic acid because it can give up a hydrogen. I think this is aspartate. Don't quote me on that. I'll look it up later. I'll let you know. But anyway, we've got our essential carbon, we've got our amino group, but this time it's charged, so there's kind of just a positive charge around that amino end with all those hydrogens that have a bit of a positive charge on each of them. This one is got the negative charge over here on this carboxyl group, but because we had a carboxyl group on the bottom, we also have a negative charge there. So there's 3 charges to consider here. We've got a negative charge because of this carboxyl group we got a positive charge from this amino group. We've got a negative charge from this carboxyl group. Now, next week when we learn about how amino acids are connected together to make proteins, we're going to see that this carboxyl group is going to be connected to the amino group of the next amino acid in the protein chain. And so it's only at the very far end terminus, the amino terminus of the protein, that you have a positive charge. And then at the very other end of the protein, the amino acids connected together, you're going to have a negative charge. And then, so overall, the hydrogen that I had here can be accounted for because it's over here. But when I said this reaction will drop off a hydrogen or a proton on the way, that is because we've got another proton to consider that lost the tug of war with an oxygen here. So 2 negative or a positive charge and 2 negative charge, and so then the total sum charge for this one is -1. And our bodies in the cells, as I mentioned earlier, are typically pH 7.4 within a cell, and so at that pH often has to do with the concentration of, well, it depends on what definition of pH you want to go with, but protons floating around is pH, and so you can imagine if the pH changes, it'll change the concentration of hydrogens, and that will change if you go to extremes in your pH. That would change if we go to more and more acidic conditions. The proton concentrations would be higher, and you might get to the point where these carboxyls would accept a hydrogen on the proton back. But anyway, that's a little bit of an advanced topic, but I just wanted to point out that the main thing you need to know is this is not what the aspartic acid, amino acid, would look like. It would be more like this. Okay, and then that's it for functional groups, although we're going to draw them many, many more times. But that's it for the lecture on functional groups. And so next week when we meet up in class again, we're going to be talking about biological macromolecules. And 3 out of 4 of the macromolecules we're going to learn about form what are known as polymers. Poly means many, and mer means parts. So these are going to be, more often than not, just stretches of parts that are connected together. Occasionally, you can have some branching, and that's the case with carbohydrates, but the nucleic acids and the amino acids are going to, for the most part, be straight. Okay, now we got polymers. Where did they come from? Well, if you've got many parts, you must be able to have just one part. And then if you connect one part to another part, you have 2 parts, which is called a dimer. When you get to a trimer, you've got 3 parts connected to each other. A tetramer has 4 parts. And then this kind of, I'm sure a pentamer is there, but at some point we're going to get tired of trying to specify how many parts it has. So if we just want to mean a few or several parts, we would say oligomer. And if we mean many parts, then we're going to say polymer. And so these biological macromolecules that we're going to learn about are quite large molecules, and it's because they have a bunch of monomers connected together, either in a straight line or some branching. So that's it for the lecture today. Thanks to my Russian friends for dropping in. As always, like and subscribe, or not.